Hey guys, so this video is on disease patterns and identification. It's one of our last lessons on biotechnology. So the learning intention of this video is to understand how disease spreads, understand how we identify different diseases and how we might treat them depending on what they're identified as. So first of all, communicable diseases are those which can spread from person to person. And an epidemiologist will study these diseases and they've classified them in four major categories. So diseases are either categorized as outbreaks, endemics, epidemics, or pandemics. And I'm sure they're words you've all heard over the last couple of months. So outbreaks are diseases which occur in prevalence at a local level. So for example, a Legionnaire's outbreak at a shopping centre often occurs because of poor air conditioning. So it's at a very local level. It's an outbreak that occurs within that small geographical region. Endemic is a disease which is present at constant low levels in a population. So it's always there, but it's never at a level that's high enough for massive alarm. Something like foot and mouth disease occurs in Australian toddlers at a low level, but it's constant. So it's always there in some form or another. An epidemic is a disease which spreads rapidly through a nation or geographical region, and it usually affects a large number of people. So for example, the cholera epidemic in India affected their drinking water, caused really, really bad diarrhea, but was contained sort of within that area, India and those surrounding countries. Pandemic, which we've all heard of, is obviously a disease, spread of disease throughout the world. Something like Zika virus or HIV have been considered pandemics, things like plague and Spanish flu. Those sort of things are ones that affect more than one geographical area across the globe. Obviously, spreading the disease is really important to make sure it's minimising its impact. So it's protecting countries, protecting industries, protecting people from that infectious disease. So there are many ways to stop diseases, things such as isolating the infected. So once they've got the disease, separating them from others. Quarantine, which prevents diseases from entering in the first place. In Australia, we're quite lucky to have a very strict vaccination schedule. So making sure that people are following that schedule to ensure that they're actually protected. And that obviously then leads to things like herd immunity, hygienic practices, so washing hands, ensuring that we go to the bathroom and clean our hands properly, those sort of things. And last of all is the actual infrastructure in place. So cleaning of streets, sanitation systems. Again, we're quite lucky in Australia that these things happen. But if you go overseas, these infrastructures aren't always in place and disease can spread quite easily. Things like clean water as well also will limit the spread of different diseases. So where do these diseases come from? So more often than not, new diseases are caused by what are called zoonoses. And these are diseases which are transferred from humans to animals. So you can see in the table on the right, we've got a wildlife circle, a livestock and a human. Now, at times they can overcross and spread from one another. So for example, right in the middle, we've got diseases such as the plague, rabies, the avian flu, that are spread between different species, whether it be livestock or wildlife into humans. Another way that diseases arise is when they're actually new strains of previous diseases. So for example, each year we have to get a different flu shot because there's a new strain of the influenza virus every year. And so that's where we start to get some of these new diseases from. And now if a population is unprepared, there's no natural immunity against that disease, then we can see that a new disease can spread pandemically and quite easily. So one of the jobs of scientists is to actually identify diseases and be able to know what disease they're dealing with. So there are three main techniques that scientists rely on. They're going to look at immunology, genetic methods, and morphology. These three methods help them to decide what disease they're actually dealing with and then how to treat that in the first place. Then immunology, what we actually do is we test for an immune response. And so if there's an immune response, it often indicates that there was an indication of an infection in the first place. And so we use these things called agglutination tests. But basically, we combine some antibodies that we know will be against a certain disease with a blood sample. And if these antibodies start to link together, then we know that maybe there was the antigen or disease present in the first place. So we're basically testing is, does our body have the antigens present, or have we made the antibodies in the past that would actually help us fight off the disease? Genetic method, so we're using some of those processes we've talked about where we will take some DNA from a pathogen, we'll start to amplify it using PCR, and then we start to compare it using gel electrophoresis. So we're going to try and identify what disease it is by comparing it against known diseases using PCR and gel electrophoresis. So it's basically a way of profiling the DNA of certain diseases and comparing them to others. And lastly, we look at morphology. So many pathogens, and it's mainly fungi and bacteria that scientists compare. have unique shapes, or they have a unique colony shape, and they grow in certain conditions. So scientists with an unknown pathogen will try and decide, okay, is this shape like anyone we've seen before? Does it have a particular colony shape that we know of? Does it grow in certain antibiotics and not others? That gives them an idea of what sort of disease they're dealing with. And all those different combinations of morphology will help them determine what type of disease. Now, once the scientist knows what type of disease they're dealing with, that leads to treating diseases. And that obviously can be treated in many different ways. So sometimes we'll use broad spectrum treatments. So things like broad spectrum antibiotics will actually impact many bacteria at once. And it's not very specific, but we're hoping that we're putting these antibiotics that we're going to knock out something we want to. Otherwise, we use treatments that might inhibit the growth or the replication of a pathogen, or others will just straight out kill that pathogen. So obviously, depending on what disease they're dealing with, scientists will attack it with a different form of treatment depending on how it behaves. So once we know, we're going to hit it with a much more specific treatment that could, I guess, eliminate just that bacteria or that virus or that fungi and not others. It does depend on how it's been identified. In review, we know that disease spreads in those 4 major patterns, and the way we can prevent these disease spread takes many different shapes and forms. So we talked about things like washing hands and sanitation, etc. So these diseases, many of them come as zoonoses, and they spread from animals to humans. And scientists rely on identifying them using immunology, genetic methods, and morphology. So once they've identified, the scientists can then treat that disease in either a broad way or can be highly specific. So I hope this video wrapped up all of our disease work. I know there's a lot to take in, but we're going to spend a few more times in class going over this stuff.